Hello once more and welcome to the last episode of our class on the respiratory system. Okay, today we're looking at what's it called? Kreb cycle. Okay, and of course, do not forget that this tutorial, this lesson, these episodes are brought to you by the O3SCHOOLS JAMB CBT practice application. If you have made a decision that you will never write JAMB twice in your life, please get the application for whatever cost. Get it and start using it immediately for your exam. We'll talk about the app in a moment. Let us see a jump pass question from the app. This is a 2021 biology question from question 17 from this application. It says the role of ATP includes all the following activities except A says the provision of energy to do work in living organisms. B says the transmission of nerve impulses. C says keeping the body warm in homothermic animals. D says producing water during aerobic respiration. All right, so we'll come back to this question and many more. Just our way of doing things, because at O3SCHOOLS we prefer to give you value first. We know, say, if we don't teach you, answering question will be the least of your problem. All right, so please, you must seek to learn first, to know. If you don't know, there's no way question will help you. Once you understand what you are being asked upon, then answering it or choosing the right answer won't be a problem for you. So please download the application, all right, and activate it. The app has all the past questions for all your subjects, all right. As you can see under the practice for UTME, if you open the app, click on practice for UTME, you'll see all the past questions from the app. All right. Also, there's a classroom on the app that has class notes. All your class notes for all your subjects are present in the classroom. There's a JAMB syllabus in the classroom, and then you can create a study plan and crash all of your JAMB syllabus for all your 4 subjects, okay, and finish it within the shortest possible time. There's a question search feature. You can see all the features being shown. All right, there's a question search feature where you can search for question according to topic. And likewise, there's a mock exam on the app every Saturday where you can compete with your mates to win prizes and, of course, to get used to JAMB pass questions, JAMB re-exam questions, as well as what? Familiarize yourself with how the examiner set their questions. All right, so please, the app is an embodiment of success. Okay, please get the app, activate it. Activation fee is not up to the price of rice and chicken in a good restaurant. Okay, 1 liter bowl of ice cream is more expensive than the activation fee. All right, so please prioritize your future. Okay, and start preparing for exam immediately. So let us go to the Kreb cycle. Okay, we'll look at the Kreb cycle, then we'll double also talk about fermentation a little, okay, and then we'll be done with respiration. All right, so what do we call the Kreb cycle? Okay, we say the Kreb cycle is the power plant of the cells. Power plant, we say Kreb cycle is the power plant for your cells, okay? Power plant is the simplest way to explain it, okay? It's the power plant for your what? For your cells. So what is the main function of this Kreb cycle? Okay, the main function is for you to what? To take in fuel. Okay, its main function is to take in fuel that comes from food. To take in fuel that comes from food, and then what? After what? Taking in that fuel, okay, it takes it and it releases what? Energy. Okay, that's the main function of what? Of the Kreb cycle. All right, it takes in fuel, okay, that comes from food, okay, to release what? To release energy. Okay, and of course, the fuel it takes is called acetyl coenzyme. Okay, so this is called acetyl CoA. Acetyl CoA, as you can see very well here, acetyl CoA. All right, so it takes in what? Fuel that comes from food called what? Acetyl coenzyme, and then what? It breaks it down to release what? Energy. It breaks it down to release energy. Okay, so now, first of all, before we get to the Kreb cycle, okay, it all started from what? From process of glycolysis. We say glycolysis is the what? Is the breakdown, okay? It's a series of reactions, okay? Lot of metabolic or pathways, okay, that leads to the what? Breakdown of glucose to what? To form what? What do we call it? Pyruvic acid. We're in the last class, okay? If you have not watched the last class, you have no business here. All right, so breakdown of what? Of glucose, series of reactions that involves what? The breakdown of glucose to form pyruvic what? Acid, okay, or I can call it what? Pyruvate. Okay, so now this Kreb cycle is also called citric acid cycle. It's also called citric acid cycle. Citric, C-I-T-R-I-C, citric acid cycle is also called tricarboxylic acid cycle. Okay, it's also called citric acid cycle, and it's also called tricarboxylic acid cycle. All right, so please take note of them. All right, so we'll say that what it takes in energy, it takes in fuel in form of what from food. Okay, it takes in fuel from food that we take in. Okay, called what acetyl coenzyme. It breaks it down, then releases what energy. Okay, and then it is stored. Okay, this energy is stored in form of molecules called what? NADH. All right, it's also called, it's stored in form of FADH. Okay, this NADH is called nicotinamide adenine. The full meaning is nicotinamide adenine dinucleotide. Okay, is what? If you see this H here, it means what is the reduced form. Okay, all right. So it's nicotinamide adenine dinucleotide. Mind you, there are a lot of things I will not talk about. Okay, because it's above what the scope of this class. Okay, but just we're going to just use what this. Don't mind the diagram. Also, just want to use to what make you have an idea of what's happening in the Kreb cycle. Okay, so this is this. So the energy is stored in what form? What these molecules, okay? NADH, FADH, all right? And at the end, okay, of one cycle, what you usually get, okay, from the Kreb cycle, we get what? NADH, we get FADH, okay? We also get ATP, which is what we're really looking for, and they also get what? CO2, which is what? Waste what material at the end of what? Of one cycle of what? Of a Kreb cycle. So when you go from here to here, all right, this is what is produced. This 4 is what we get. All right. So now, let us proceed. So we have said already from glycolysis that glycolysis involves series of reactions. Okay, where what glucose is being broken down to what? To pyruvic acid. Now, the pyruvic acid has how many molecules? Okay, how many carbon atoms? The glucose itself has 6 carbon atoms. It's a 6 carbon atom, isn't it? The glucose is a 6 carbon atom, and then it is broken down to pyruvic acid, which is a 3 carbon atom. All right. So now it's broken down to a 3 carbon atom, and this pyruvic acid wants to enter into the mitochondria, which is what the powerhouse of the cell, so that it can enter into the what? Into the Krebs cycle. But the pyruvic acid cannot just enter into what? The mitochondria, because of the mitochondria, okay, has what? Inner and outer layer. Okay, or inner and outer membrane. So it cannot just enter into the mitochondria like that. So it has to be converted. It has to be transformed, okay, into what? A 2 carbon atom. A 2 carbon atom called what? This acetyl coenzyme. All right. So this is what happens. This coenzyme, CoA, when it is added to carbon chains, what it does is that what? It transports what? The carbon molecule and facilitate the carbon molecule by being utilized by an enzyme. So it can now what? Go into what? Into the Krebs cycle. So this is our acetyl coenzyme. All right. So this is the first state. This acetyl coenzyme is what is first of all what being what? Converted into what? Citrate. CITRATE, as you can see very well here. Okay, it is being converted to what? To citrate. CITRATE. Now, what happens here? The conversion, okay, of this acetyl coenzyme to what? To citrate. Okay, there's an enzyme that does this. Okay, but first of all, this acetyl coenzyme, okay, is what? Being what? Combines with what we call oxaloacetate. Okay, it combines with oxaloacetate to now give us what? This what? Citrate. Okay, and the enzyme that does this, the name of the enzyme that does this, that is conversion from what? This acetyl coenzyme to citrate. Okay, the enzyme is called citrate synthase. Okay, the enzyme here is called citrate synthase. Okay, let me find somewhere to write it. The enzyme here is called citrate synthase. Okay, that's the enzyme that helps in the conversion of what? Of acetyl coenzyme to what? To citrate. Okay, and then moving further, remember first of all that what? Your acetyl coenzyme is what? How many carbon? This has what? 2 carbon. Okay, and then of course your citrate definitely what? Has what? It has 6 carbons. Your citrate has 6 carbons. Okay, your citrate has 6 carbons. Okay, please take note of that. Then now, let us now go. So from citrate, it goes to what? To isocitrate. Okay, from citrate, it goes to iso what? Citrate. Okay, so I've seen the enzyme here is citrate synthase. Okay, now from citrate to isocitrate, okay, the enzyme that is responsible for this conversion is called aconitase. Remember, your isocitrate is also what, 6 carbon. Okay, the enzyme here is what, is aconitase. Please take note of that. All right, so from acetyl coenzyme to citrate is what, citrate synthase. From citrate to isocitrate is what, aconitase. All right, then now the next stage is what, is the movement, is the conversion of what, isocitrate to what, to alpha ketoglutarate. Okay, isocitrate to alpha ketoglutarate. Alpha ketoglutarate here has what, 5 carbons. Okay, it's 5 what, that's what, it's a 5 carbon what, compound. Okay, so alpha ketoglutarate is being what, from isocitrate to alpha ketoglutarate. Okay, the enzyme that is involved in this is called isocitrate dehydrogenase. Okay, isocitrate. Okay, look at this, it's called isocitrate. Okay, that is this isocitrate, then dehydrogenase. Okay, and this process produces what, NADH, as you can see very well, produces what, NADH, and also produces what, CO2. Okay, which is a waste material. Okay, your CO2 is a waste material. All right, so please take note of that. So from acetyl coenzyme to citrate is citrate synthase. From aconitase, sorry, from citrate to isocitrate is what, aconitase. From isocitrate to what, to alpha ketoglutarate is what, isocitrate dehydrogenase. Okay, having what, your alpha ketoglutarate has 5 carbons. Okay, then the next stage is what, is the oxidation of alpha ketoglutarate to what, succinyl coenzyme. Okay, this is the next stage. Okay, the conversion of what, or oxidation rather, of what, of alpha ketoglutarate to succinyl coenzyme. All right, and this process also produces what, NADH. It also produces NADH and what, and CO2. Okay, please take note of that. It also produces NADH and CO2, and the enzyme that facilitates this conversion is called alpha ketoglutarate dehydrogenase complex. Okay, please take note of that. Okay, so the enzyme that facilitates the conversion of what, alpha ketoglutarate to succinyl coenzyme is what, alpha ketoglutarate dehydrogenase complex. Okay, please take note of that. So the next stage now is what, is conversion, okay, of for this succinyl coenzyme, okay, to what, to succinate. Okay, conversion of succinyl coenzyme to succinate. This process produces what, ATP or what, GTP. Okay, produce what, ATP or what, GTP. Okay, so the phosphate present in what, it's what, transferred, okay, to get what, your ATP. All right, so we get what, our ATP here. All right, in this stage, and the enzyme that is involved, okay, in the conversion of this succinyl coenzyme to succinate is called succinyl coenzyme synthetase. Okay, succinyl, look at this, succinyl coenzyme, okay, coenzyme A or CoA, okay, synthetase. Okay, please take note of that. Okay, so the enzyme that is involved in the conversion of succinyl coenzyme to succinate is succinyl coenzyme synthetase. Okay, and this process leads to what, production of what, ATP. Now that will take us to from succinate to what, to fumarate. Okay, from succinate to what, to fumarate. So succinate, okay, is what, is oxidized to what, to fumarate. Okay, succinate is oxidized to what, to fumarate. All right, and in this process, FADH, okay, FADH2 is what is produced. That is FAD, okay, that's what reduced, the reduced form of FAD. Okay, so that is what FADH. It is produced in this process, and the enzyme that is involved, okay, in the conversion or in the oxidation of succinate to fumarate is called succinate dehydrogenase. Okay, this one is called succinate. Where should I write this? All right, this one is succinate dehydrogenase. Succinate dehydrogenase. Okay, so that is the enzyme that is what, responsible for the oxidation of succinate to what, to fumarate. So from fumarate, it goes to what now, to malate. Okay, it's being converted to what, to malate. From fumarate, it's been converted to what? To malate. And fumarate, through the process of what? Of hydration. Through the process of hydration, hydration, Hydration. Okay, hydration is what? Is the addition of water. Okay, it's not dehydration. Dehydration is removal. Hydration is addition. So through the process of addition of water, okay, fumarate, through the process of hydration, fumarate is converted to what? To malate. Okay, and of course, this process, as you can see here, water has been added, and then what? The enzyme that is responsible for what? For this hydration is called fumarase. Okay, it's called fumarase. Fumarase. Okay, then of course from malate it is being converted to what? To oxaloacetate. Okay, we are drawing closer. Okay, so from malate it is being converted to oxaloacetate. So here malate is being oxidized. Okay, oxidation happens, it's being oxidized to what? To oxaloacetate. Okay, it's being oxidized to oxaloacetate, and of course, as you can see vividly here, okay, NADH is what is produced. Okay, and of course, the enzyme responsible for this is called what? Malate dehydrogenase. Okay, malate dehydrogenase. Okay, and of course, obviously, oxaloacetate is what is reproduced, and then what? The cycle what continues. All right. So it combines again with what? With acetyl coenzyme. Okay, to form citrate. Okay, it continues like that, and like that. That's why it's a cycle. Okay, it's an endless loop. It continues. All right. So this is the Krebs cycle. Okay, in a nutshell. Now, the net product per acetyl coenzyme. Okay, that is 1 ton. Okay, 1 ton of the cycle. Net products. Okay, let us see the net product. Net products per acetyl coenzyme. All right, net products. Okay, per acetyl coenzyme, that's what? 1 cycle. Okay, 1 ton of the cycle. All right, so we are going to have a 3 NADH. All right, we have 1 FADH. Okay, we have what? 1 ATP. Then we now have what now? 2 molecules of what? Of CO2. Okay, so in 1 ton of the cycle, this is what is what? This is what is produced. Okay, but remember that what? 1 molecule of glucose, okay, produces what? 2 molecules of what? Of acetyl coenzyme. Okay, 1 molecule of glucose produces 2 molecules of acetyl coenzyme, and for that reason, that means that the total what? Net product that we produce will be what now? Times 2 each. I'm going to have 6 NADH, I'm going to have 2 FADH, I'm going to have 2 ATP, okay, and I'm going to have what now? 4 CO2. This CO2 is what? It's a waste material, so it's what? It's let out, it's expelled out of the body. Okay, so this is the Krebs cycle in a nutshell, right? So now I want to quickly look at fermentation. So now if someone asks you what is the significance of this Krebs cycle, okay, what is even the usefulness of this Krebs cycle, you should be able to what? At least say 1 or 2 things. What are those things you should be able to say? Number 1 is that what? That the Krebs cycle provides electrons. Okay, it provides what? Electrons. That is what? Your nicotinamide adenine dinucleotide, as well as your flavine adenine dinucleotide. Okay, which helps in what? In electron transport chain. Okay, it helps in what? Electron transport chain. So number 1, you can say what? It provides, the Krebs cycle provides what? Electrons. That is what? Your NADH and FADH for the electron, okay, transport chain to generate ATP, which is what you are really looking for. Okay, so provides electrons, that is your NADH and FADH for the electron transport chain to what? To generate what? ATP, adenosine triphosphate. Then likewise, number 2, it supplies intermediaries. And say it supplies intermediaries for biosynthesis. Okay, for example, your amino acids. Okay, it provides what? Intermediaries for biosynthesis. Likewise, you could also say that it regenerates oxaloacetate so that the cycle can continue. Number 3, it regenerates oxaloacetate for the cycle to continue. Pardon. So now, this is what it's not in a nutshell, all right? So now, quickly, let us quickly touch fermentation, and we'll know that we are done with what? With respiration, okay? So what is fermentation? You can see fermentation as the anaerobic respiration of yeast, okay? And define what fermentation as what now? Anaerobic respiration of yeast. Now, in this respiration, okay, glucose is what? Is converted to what? To pyruvic acid. Glucose is converted to pyruvic acid, okay, and then pyruvic acid is converted to what? To ethanol, all right? So that is what fermentation that we're looking at, okay? Fermentation, okay? The anaerobic what? Respiration of what? Of yeast. That is what fermentation is what it's all about, okay? Saying this respiration, glucose is converted to pyruvic acid, and then pyruvic acid converted to what? To ethanol. Then we also have the one we call lactic acid fermentation. Lactic acid what? Fermentation, okay? All right, so in lactic acid fermentation, okay, glucose is first of all converted to pyruvic acid, okay? Then the pyruvic acid is now converted into what? Into lactic acid by what? NADH, by NADH, all right? By your nicotinamide adenine dinucleotide, okay? It is being converted first of all from glucose to pyruvic acid. So for lactic acid, it's glucose to pyruvic acid, okay? Then from pyruvic acid, it is now converted to what? To lactic acid. Lactic acid by what? By this what? Na or DH, okay? Be converted to what? To lactic acid without the production of CO2, okay? Without the production of what? Of carbon 4 oxide, okay? So this is lactic acid what? Fermentation. Take note of that, all right? So for anaerobic respiration of yeast, all right, we get what? Glucose be converted to what? To pyruvic acid, and we get what? An alcohol, all right? We get an alcohol, and we get what? Carbon dioxide or carbon 4 oxide, all right? But for lactic acid fermentation, glucose is converted to pyruvic acid, from pyruvic acid converted to lactic acid, all right? By what? By NADH, this nicotinamide what? Adenine dinucleotide, and then what? CO2 is not produced. Take note of that, all right? Check your past question, you see them, all right? Take note of that. It's very, very, very useful and important. I would have told you if it was not. And in alcoholic fermentation, take note, in alcoholic fermentation, okay, note that what? Only 2 molecules of ATP, okay, are produced per what? By 1 what? Molecule of what? Of glucose, okay? Take note of that. Only 2 molecules of ATP are produced in what? Alcoholic what? Fermentation, all right? Take note. But in aerobic respiration, okay, you know that what? 1 molecule of glucose produces how many ATP? Produces 38 ATP. You see it in your past question, all right? Please take note of that, all right? So in alcoholic fermentation, or the one we can call anaerobic what? Respiration, okay? We'll call it anaerobic respiration of yeast, okay? It produces what? 2 molecules of what? Of ATP. But this aerobic respiration produces 38 molecules of ATP. So let us go to past question, answer questions, and we are out of here. The first question here we are having from 2021 number 17. It says the role of ATP include all the following activities except say provision of. You know, there are some questions that even if you don't know the answer, you can use your sense to just get it. This is one of them, all right? A says provision of energy to do work in living organisms. Yes, ATP does that because ATP is what? The energy currency, okay? So it's what? Produces what? Energy for many what? Activities. Transmission of nerve impulses, you need energy also. C, keeping the body warm in homothermic animal, you need ATP. D, producing water during aerobic respiration. Does that seem like an answer that would be correct or wrong? Okay, so correct answer is option D. D is not a role of what? Of ATP. Okay, so let us see 2001 number 8. It says 2001 number 18. Sorry, 2001 number 18. It says when yeast respires anaerobically. When yeast respires anaerobically, that's why I say anaerobic respiration of yeast is fermentation. Okay, it says convert simple sugar to carbon 4 oxide and A say acid, B say water, C say oxygen, D say alcohol. Correct answer is alcohol. Okay, so a product, okay, of the anaerobic respiration of yeast is what is alcohol. Alcohol is part of them. CO2 is what is part of that. All right. And of course, we said what 2 molecules of what of ATP is released. But for lactic acid fermentation, CO2 is not produced. Please take note. You can they get you with that kind of question. Please be very, very careful. 2020 number 28. You answer this one. Say the process of anaerobic respiration of yeast in sugar solution is known as. Okay, just tell me the correct answer in the comment section. Thank you for joining in. My name is Master T, and I'm my humble self. And I'll see you in the next class. We'll start discussing excretion. Thank you for watching.